next, the cell biology lab will be represented by Nikki Jacobson, Nathan Landry, and Ian McAdams. The title of their talk is Examining Expression of ATG1 homolog candidate genes in autophagy-induced tetrahymenate Thermophila. Yeah, let's see if this behaves a little bit better.
Here we are. Hi guys, I'm Nikki. That over there is Nathan, and this is Ian. We're from MCDB 3140, which is the cell biology laboratory. And today I'll be talking to you about examining the expression of ATG1 homolog candidate genes in autophagy-induced tetrahymenate Thermophila. So the cellular process that our lab looked at was autophagy. And autophagy is this basal cellular process that's kind of at a constant level in normal healthy organisms. It's involved in cellular growth, aging, as well as cellular remodeling. However, there are certain situations such as stress or starvation that can cause autophagy to be upregulated or for it to increase in the cell. And when that happens, the cell has a little bit better of a chance of surviving that stressful period. There are also a lot of different diseases and dysfunctions that involve autophagy, as shown by that wonderful image there. One good example is cancer. Cancer is one of these weird ones that you can either have the upregulation or downregulation of autophagy. Another example is Parkinson's disease or other neurodegenerative diseases in which plaques are formed by the buildup of proteins. So in this case, autophagy is downregulated, so we're not breaking up those proteins that should be being broken up. Here's just kind of a brief overview of autophagy. We first have this isolation membrane, also called an autophagosome, and it isolates the different components in the cell that are being targeted for degradation. We then have the lysosome, which carries proteins and enzymes that degrade the things, and they merge together, and then everything's broken up, and the cell continues on. So we have this little guy here, TOR, called target of rapamycin. I have problems with that word. And it hyperphosphorylates ATG13, which then prevents the ATG complex from forming, and then prevents the autophagosome from forming, and then autophagy can't happen. But when a cell is starved or stressed, TOR is inhibited, which allows ATG13 to complex with ATG1, which then forms the ATG protein complex, and then makes our autophagosome, and then autophagy is induced. Autophagy is conserved across evolution and is conserved across eukaryotes such as platypuses, birds, amoebas, as well as humans, and our organism Tetrahymenate Thermophila. So this is really cool because we can do experiments in these organisms and then apply that information back to us.
So tetrahymenate is a single-celled eukaryote. It's found in freshwater environments. It has nuclear dimorphism, which means it has two nucleosomes, so one macronucleus and one micronucleus. Its notable discoveries that has been utilized in was research for ribosomes and ribozymes and telomerase activity. Now, for our lab, it had four primary aspects that led it to be our primary model organism. And the first and foremost for this type of environment was that it's very easy to both grow and maintain, as well as being very inexpensive to work with. So it was perfect for all of our various work groups to utilize. Next is the aforementioned dimorphic nuclei. So the macro and micronucleus allows us to independently and separately and specifically modify one or the other in order utilizing homologous recombination to either modify, remove, or add specific genes of interest. Also, due to its large translucent structure, it's very easily visualized in a lab environment. And then lastly, like Nikki mentioned, it contains several conserved eukaryotic processes, like autophagy, that make it a great parallel for research with other organisms, including humans. Some other organisms that Tetrahymena will hopefully function as a possible model organism for are those like Plasmodium falciparum, responsible for the disease malaria, and Toxoplasma gondii, responsible for the disease toxoplasmosis. So it's hopeful that Thermophila, being an alveolate like these two, can contribute to their research as well. All of this ties back into our overall question for this lab. Do our selected genes function as ATG1 homologs in Tetrahymena thermophila? So the first thing each group had to do was select a gene. Our group selected the gene TAC20, which stood for Tetrahymena autophagy candidate number 20. Our gene was 2,429 base pairs long, had one internal intron, and then 4 other sequences of interest. These were serine threonine kinase, 2 coiled regions, and this purple one here was our singular small low complexity region. After selecting our gene, we then had to design a primer in order to actually replicate and examine in bulk our gene of interest. So what we took advantage of was our intron. So by designing our primers to flank this intron, we were able to make sure we could control the differentiation between cDNA and gDNA, which allowed us to rule out a common contaminant, which would have been gDNA products. Because gDNA expression does not change, it allowed us to make sure we could control which one was which. After we had designed our primers, we were then able to move forward with the standard experiments. The first of these was by taking Tetrahymena thermophila samples and exposing them to a hydrogen peroxide concentrated solution of 10 micromolar, and this was done for just over an hour. The purpose of this was to stress the cells in the hopes of inducing autophagy, which would hopefully force upregulation or downregulation of genes involved in autophagy. For example, our ideal ATG1 homologs. After stressing the cells, we lyse them in order to get at their internal components and therefore isolate our mRNA. With that isolated mRNA, we then ran an RT-PCR to produce cDNA. And with this cDNA and primers, we tested them for functionality, confirming that we were able to move forward from this stage. With those two, we then ran a PCR assay, as well as utilizing S2018 proteins. And after all this, we got to the best part, where we were able to take all the PCR products and visualize them by running an agarose gel electrophoresis process on a 2% concentration agarose gel.
So here are the results from our first PCR assay. We used TAC20 primers and 4 distinct cDNA stocks. Our stressed cDNA stocks are noted on the slide in red, and our unstressed cDNA stocks are noted on the slide in yellow. The unmarked lanes are our controls. We used HHP1 to ensure that our cDNA wasn't degraded, and we used our TAC20 primers with gDNA to make sure that our TAC20 primers were still working. The only notable result on our first gel is circled over there in the big yellow box. We had some unstressed cDNA plus our TAC20 primers show approximately less than 10 nanograms of product. Because of the low amounts of product and because this result wasn't repeated throughout the gel, we were kind of leaning towards this was contamination at this time. But how do you test a hypothesis? You run another experiment. And here's our second PCR assay. Again, our unstressed cDNA is marked in yellow, and our stressed cDNA is marked in red. So our unstressed cDNA here in lanes 1, 2, 8, and 9 all produced around 50 nanograms of product in each lane, while our 3 and 4 with stressed cDNA produced no product. And the most interesting result here was our stressed cDNA showed less than 10 nanograms of amplicon. So I don't know if you guys can see that, but it's there, I promise. Right there in that box down there on lanes 10 and 11. Again, because these results weren't really consistent and we had such a low amount of product, we were beginning to suspect that there might be a contamination problem. So moving forward for our conclusions, we don't really know if TAC20 plays a role in tetrahydrothermophila autophagy. We had no amplicon and then amplicon in unstressed cDNA, and we also had amplicon and then we had no amplicon in unstressed cDNA. So our results were all over the board, and we can't really draw too many conclusions from it. And our research question requires further experimentation. So for our future directions, we'd like to examine our upregulation of TAC20 over a time gradient. So what that means is basically we'd like to expose our cells to 10 millimolars of H2O2 and then see if there's any gene expression at 10 minutes, 20 minutes, 30 minutes, all the way out to an hour, like our initial experiment was. If we do note upregulation over our time gradient, we're going to tag our protein TAC20 with a GFP tag and see if it localizes with the autophagosome to further show whether or not TAC20 plays a role in tetrahydrothermophila autophagy. Well, I guess I'll just finish it out then. If we don't see any upregulation over a time gradient, then we're going to, we'd really like to see what TAC20 does. So we're going to try and hit the tetrahydrothermophila with a temperature gradient and see if there's any sort of expression there. Put it under conjugation conditions and see if there's any upregulation of TAC20 under conjugation, and then infect the cells and see if we can get any TAC20 amplicon out of that. Does anybody have any questions? So the question was, how would we visualize the autophagosome to make sure that the protein and the autophagosome are together? There are dyes that you can use. The autophagosome is a structure. It's like a big double membrane brown structure. And if you stick some dye into the cell eats it, puts it in the autophagosome. And so you have the GFP tag protein and the autophagosome. You can visualize them both with certain things that I can't remember at this time because I'm super nervous. Does that answer your question? Any other questions?
So just to reiterate, her question was, is there any evidence that mRNA expression evidence has been seen with regards to ATG1 expression as opposed to what, N protein product? Is that correct? Right. There could be. I've not seen any studies saying one or the other up to this point. I'd have to look into that and get back to you.
So just to add to that real quick, we're looking at the final product. This is the product that's printed out to make the protein that, the mRNA is the product that's printed out to make the protein that forms together that's supposed to make the autophagosome. So yes, it is already like cut up and all the introns are removed. Is that your question? So why did we choose these genes as ATG1 candidates? Is that right? So it's a serine threonine kinase, and we grabbed a bunch of serine threonine kinases and we just kind of ran them through brute force to see if they were ATG1 homologs.
So we're also basing our, we're looking at ATG1 because ATG1 is the protein in yeast that has been studied, and it's mostly, like, we're basing this also of studies in yeast. We don't really understand it in tetrahymena. So that's why we're looking at the ATG1 gene, because in yeast, we know that ATG1 is what kind of pushes autophagy forward. So we're taking that information from yeast and seeing if we can find similar circumstances in tetrahymena thermophila. So we don't know specifically if there are any like post-translational modifications that happen in tetrahymena thermophila. We're just seeing if there's a gene similar to ATG1 that we found in yeast in tetrahymena thermophila, if that answers your question. Okay, sorry.